Okay, so let's go deeper. So it's this area will be the connective tissue. Okay? So you will see a lot of components here. So there are key extracellular matrix components in the connective tissue will be phytopathology, GAG, which is repeat disaccharide 2 sugar, and proteoglycan is a protein glycoprotein contained GAG disaccharide. Okay, and the multi is a protein like glycoprotein and elastin. Okay, so those ECA components are mainly produced by the fibroblasts. Okay, there are few fibroblasts inside here. They will keep general. They actually fibroblasts right here, not here. Okay, fibroblasts here will generate a lot of those components here. Okay, and gradually form a big connective tissue. So, first, as I mentioned last week, the cell process is really crowded. So, the space is really limited, although they draw nothing in the cytosol. But actually, in the cytosol, it's super crowded. The molecule bigger than 20 nanometer in diameter, they cannot reproduce. Okay, but those extracellular matrix, as I mentioned before, they are synthesized by the protein. They need to synthesize protein from the ribosome, and they need to secrete out those components to the extracellular space. And those extracellular matrices, they are huge. How can protein synthesize huge protein and kind of deliver them in such a crowded cytosol environment? That would be my question. Okay. For example, collagen is easily, this is the size of the collagen fiber. It's huge, larger than 250 nanometer. Okay. But the pore in the cytosol is only 20 nanometer. How can they synthesize in such a crowded environment? Okay. So yeah, cell find a way to deal with this. So of course, this is the mechanism for cell to synthesize collagen. There are several ribosomes will bind to the. Do you know what kind of factor determine which ER is smooth ER, which ER is rough ER? There's different two kind of ER, SER and RER. Who determine? Ribosome. So ER with ribosome would call raw ER. And the ER without ribosome will be smooth ER. And the smooth ER will be the one, the RER, the major function is to synthesize protein. SER, the major function is to synthesize lipid. That's what you learn, right? And actually, the upstream factor will be mRNA determine who will be the RER. So ribosome in the beginning, on the steady state, they are free to diffuse in the cytosol. Okay, and the nuclear mRNA will secrete out from the nucleus. Once the DNA become activated, they will kind of transcription, and then become the mRNA will go through the nuclear more complex and then go to the cytosol. And once they go to the cytosol, the ribosome will recognize those mRNA and start to read the codon on the mRNA. It is trying to conjugate with corresponding amino acid. They will generate polypeptide. Okay? If they realize those polypeptides are soluble protein, they will keep in the cytosol. And they just generate soluble protein. Then those soluble protein will freely diffuse to their destination. If they realize wow, there's a transmembrane domain, those amino acids are hydrophobic, they will attach to the ER lumen and start to synthesize the membrane protein. Okay, so mRNA determine which ER will become the SER and the RER. So you will find more RER around the nucleus, and inside the cell, and more SER in the periphery of the cell. That is because the concentration of the mRNA is higher around the nucleus. So there will be more ribosomes bind to the mRNA than bind to the ER. So there are more ER become RER around the nucleus. Okay? This is all about this clear mechanism right here. Okay? So those ribosomes will receive the collagen mRNA and start to bind to the ER membrane and generate, as I mentioned before, the collagen are trimeric. They are 3 polypeptides. So we alpha 1, alpha 1, alpha 2 in this case. Okay. And so once they synthesize the polypeptide, they will use the C-terminal modification to form the disulfide bond. Okay, as you can see here. And the glycine XY repeats, they will kind of form such a covalently modification will form such a finally will form such a helix, triple helix. Okay. And some P-sharp protein 47 some shape protein will bind to those triple helix component to prevent the larger or abnormal aggregation. Okay, so they can keep a beautiful structure here. We call this pro-collagen. Okay, unmature collagen, kind of unit of the collagen. So it's a pro-collagen. And they will use the modification on the tube side to align with each other. They will kind of in the cytosol, it's crowded, but in the lumen, it's really kind of open space, really kind of a huge space here. Okay, so they kind of will deliver to the goat. Basically, goat is also grows from ER. So ER will accumulate to a lot of goat specific enzyme specific protein that gradually form goat. So goat grows from the ER. Okay, a lot of organelles grow from ER. Okay, so those collagen will allow with each other and form such a electron association. And then the goats also have a huge lumen. Okay, then they will deliver directly to the extracellular space. After secreting out of those pro-collagen, they will take out those the end orbital domain. Then these guys, pro-collagen, kind of can serve as a unit to assemble with others in the extracellular space outside the cell. It forms a huge component here. Okay, the size of this unit, as you can see in this band here, is about 67 nanometer. Okay, and the vesicle size is usually average between the 30 to 60 nanometer diameters. So it's good to deliver collagen unit, but not good for delivering collagen fiber. So they need to synthesize and deliver the collagen unit inside cell. Once they secrete out those units, they will assemble outside the cell, and they can form such a huge extracellular protein. Okay, that's the way. As you can see here, so this is a unit. Okay, that's the way cell to deal with such a huge protein synthesis. Okay, and there are two different kind of collagen. So type 1 collagen, which usually form the fibriller type, it will form the long fiber. And type 2 collagen will usually, as you can see here, will usually form the mesh fiber network. Okay, why? Because type 1 collagen they will bind. This will be type 1 collagen fiber. Okay, they will bind with the type 4 collagen, which have this, the streak pattern. But type 2 collagen, so after a while, so those are two collagen will form the long fiber overall. But the type 2 collagen will bind to the type 9 collagen, which have the king here. Okay, they have a king in the structure. So they can kind of this king will extend the type 9 collagen to the other side and will bind to other type 2 collagen. Finally, they form the mesh pattern. Okay, so those are long fibers. Type 1 collagen you can usually bind to the some tissue called the tendon. Okay, because this will be the tendon, and then if you last year the collagen here. The type 1 collagen are all orientated in the direction of the stress applied to the tendon. The stress will be directionally in the tendon will apply here, so it will be all aligned with its stress. Okay, but the type 2 collagen, which is for the mesh, there's no orientation, just for the hue mesh here, or kind of usually exists in the cartilage. Okay, one cartilage or connective tissue, because this stress can be applied to the different direction. Okay, there will be no clear or routine direction in the cartilage. Okay, so now we talk about the GAG. So GAG stands for glycosaminoglycan, which is a non-linear polymer of the repeating disaccharide 2 sugar. Okay, it's 2 sugar. So it will be repeating these 2 sugars. So in the test code, they introduce 4 different GAG. It's a iron, ammonium sulfate, heparin sulfate, converting sulfate, and keratin sulfate. Okay, there are a lot of, as you can see, there are a lot of negative charge here. Okay, so there's a low polymer GAG will have a lot of negative charge, and negative charge will attract a lot of water here. So around here, there will be a lot of water here. So it basically will generate like a gel-like environment around those proteoglycan. Okay, that is why your connective tissue will have flexibility, but still have mechanical support. Okay, it's not a bone. Bone don't have flexibility, but your connective tissue has flexibility. Okay, it's more than gel. Okay, so the sugar, it depends on what kind of amino acid. The sugar will only bind to certain amino acid. If it bind, it only binds to the sweet plant amino acid. If it binds to the OH group of the serine and the surrounding, we call this only oligosaccharide. If it binds to the NH2 group of the asparagine, we call this only oligosaccharide. Okay. So this, for example, this will be a linkage sugar bind to the serine, we call this only oligosaccharide. After linkage sugar, they will call this repeated disaccharide. In this case, we conjugating something. Usually, it can extend 200 conjugating something. Okay, and then that is why you will see this will be the repeated disaccharide. They conjugate with certain. Those are heparin, for example. Or GAG is important for certain physiological conditions. For example, heparin. This is the blood vessel and the endothelial cell. The first line of the endothelial cell wall. Okay. The surface protein contains some this endothelial surface protein contains some heparin sulfate. So heparin sulfate, those are heparin sulfate, and it provides some SO3 groups. Okay, and the pink one is good for the 83 protein binding. So this 83 is called anti-thrombin. It will block thrombin protein's function. Okay, and this thrombin function is trying to kind of induce blood clotting. Okay, so anti-thrombin will inhibit blood thrombin function, and then will inhibit suppress blood clotting. And then binding of anti-3 with the heparin sulfate will activate NT3 and inhibit thrombin and suppress blood clotting. If you don't have no heparin, you will have inactive NT3 and have active thrombin, and then trigger a lot of blood clots. Okay. The people also recently found that heparin sulfate is really important for the SARS-CoV-2 COVID-19 virus going to the cell. Okay, those are triggers such a virus infection. Entry. Okay. Yeah. Okay. So this is the EN of the hyaluronic acid. Okay. So this yellow line is a hyaluronic. So hyaluronic, a molecule that will conjugate with some aggregate, which is one kind of protein. So this protein will also kind of serve as a branch, will conjugate with a lot of linkage sugar that is a different GAG. So you can imagine there are a lot of negative charge here and attract a lot of water here, and can form the gel-like environment here. So some kind of gel-like environment is really important. If the tissue, the extracellular space is too compact, there will be no brain space for the cell to do a lot of things. For example, the cell need to grow, cell need to migrate, cell need to proliferate, cell need to form the huge, for example, the branch pattern I showed you before. If they want to do some activity, they need space. If they are too compact with each other, they cannot do anything. Okay, so in this case, they can give some flexibility for cell to do some reaction. So it's also important for this. Okay. And also talk about the fibronectin, which is a multi-adhesive molecule. You will see again, and this is fibronectin. It's got a lot of binding site for other molecules to bind with fibronectin. It also have an RGD domain, which is good for the integrin interaction. Okay. So this is the RGD sequence, which is good for the integrin interaction. This is a experiment to prove RGD such a amino acid combination is critical. Okay, if you synthesize different polypeptide like a D, R, E, D, S, R, Z, G, K, G, D, S, P, blah, 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 different amino acid sequence, and then you incubate with coating with the culture dish with the increasing concentration of these different polypeptide on the culture dish, then you just check how many cells can kind of really successfully see on those culture dish. And then you found only polypeptide with RGD can help cell to adhere on the culture dish. Even you have R, you have a G, you have a D, but not RGD, they don't care. Even you increase the concentration of those polypeptides, the cell still can successfully adhere on your culture dish. Okay, so RGD is really important. Yeah. Okay, so this in the connective tissue, we will also find some thick fiber here. It's called the elastin. Okay, elastin can even form a bigger fiber compared to the collagen here. It also kind of can provide some mechanical support in the connective tissue. Okay, so such an ECM composition or density can need to be highly regulated. As I said, if the cell needs some space to do their activity, they need to be digested, they need to become loose, okay, right? So several enzymes, like zinc-dependent matrix metalloproteinase, those enzymes kind of secreted from the cell can digest those extracellular matrices and reduce the density of those ECM, generate more space for them to proliferate or even apoptosis or do some higher order of those tissue pattern, like those tube formation. Okay. So AMPS, ADAMS, and ADANTS are the different family of those AMPS, those enzymes to digest the extracellular matrix. And those enzymes also need to be highly regulated, otherwise they will destroy every extracellular matrix, right? So those enzymes can be also controlled by different inhibitors like TIPS and the RAS. Okay, RAS. Okay. So by doing it with this enzyme inhibitor, they can coordinate finally digest properly with those extracellular matrix for them to do the proper work. Okay. So in this part, we talk about the key ECM composition in the connective tissue. We introduce how the cell can synthesize the fiber collagen by their really crowded environment. So they basically just synthesize the unit first, then secrete out those unit, and then they will assemble the unit of the fibroblasts outside the cell. Okay. And also introduce GMG. There's a repeating assembly, and then the protein content of the GM conjugated with those GMG, we call a protein glycan. Okay. It depends on the amino acid they conjugate with. We call it olive and unlive polysaccharide. And it also fibronectin is a multi-adhesive protein which have a lot of binding sites for other proteins to bind with, and also have RGD domain for the integrated interaction, and also introduce elastin. Okay, and the three family of the metalloproteinase and the ADS and ADNTS can be modeled and integrate the ECM for cell to do the other activities, and several inhibitors can also control those enzyme reaction. Okay, any questions so far? No? Okay. So far, we only talked about the case in the static. Okay, the cell just stay there and they have an interaction with others. But it's not, the cell will move. Sometimes they are motile, okay, right? So now we kind of shift to the situation, the cell, when cell move, what kind of interface or what kind of cell-cell adhesion they will kind of establish. So as I mentioned before, the EMT epithelium essentially will transition the cell. After this EMT, the cell will become migrating type of cell, just like we working on the flow, we need to step on the flow, right? We need to have an interaction with so those are flow. The cell migration also need kind of have interaction or adhesion with extracellular. Otherwise, they cannot fly. Okay, they need to adhere there and later migrate. So during the migration, cell basically the adhesion of the cell, they are not totally rely on the bottom of the cell. Okay, they actually use some spatial structure to stand on the bottom of the culture dish or stand on the extracellular matrix. We call this specific site adhesion site. So if you enlarge those adhesion sites, you will see there are a lot of integrin protein inside this adhesion site. And those integral contribute some adapter and some active field inside here. Of course, for example, this is FAK, several adapter protein FAK here. So if you label FAK inside this is one kind of cell, you will know focal, we call this a focal adhesion complex. They have a lot of different names. Focal adhesion, I usually call it focal adhesion complex. Okay? So this will be the focal adhesion complex cell stand on the bottom of the culture dish. Okay, they are not totally designed line on the bottom. They use this different style to stand on the culture dish. Okay, and during the migration, for example, this is the direction of the cell, this cell migration. They need to form a new adhesion site, and then they can extend their cell mass. And of course, they also need to do the contraction in the other side. Then the whole cell mass will kind of move toward the destination. Okay, and in the other side of the adhesion site, they need to remove the adhesion site. Otherwise, if my cell cannot remove from there, I cannot walk further. Okay, I need to form a new step and remove the old step. Okay, then keep walking. So this adhesion site needs to be really done. Otherwise, they cannot grow. Just like how we work, right? Okay, so what happens if those focal adhesion site becomes static? This green signal will be the focal adhesion site. So this cell will keep migrating, and then you can see they will keep form new focal adhesion complex and then remove the old focal adhesion complex in the other side. And then the normal cell, you still can see the new focal adhesion complex here. If you use some treatment where you inhibit such a dynamic in this space, they cannot move. They will stay there. Okay? So, as I mentioned before, the major component of the focal adhesion complex is integrin. So if you can control, basically cell will control the activity of the integrin to form a to activate the new adhesion site and then inactivate the old adhesion site. So they need to kind of activate the integrin in where they try to form the new adhesion site, and then inactive integrin of the old adhesion site. So there are two mechanisms to control the integrin's activity. First, you just regulate the integrin's expression rate. Okay, but it doesn't make sense to use this mechanism to regulate the adhesion site because it's too slow. If the cell receives the signal and the signal transferred to the nucleus, and the DNA start to do the transcription and finally mRNA, and they go to the ribosome translation, and finally this protein will go to the process member. It usually takes 15 to half hours to do this. Okay, so if you want to regulate the integrin by regulating the expression level, for example, you want to generate a new adhesion, you need to produce more integrin. It takes longer than half an hour, theoretically, right? It's too slow, but the cell needs to migrate faster. So they will not use this mechanism to regulate such a fast event. Cell migration is kind of fast. So they use other ways. They use change their conformation. There are two types of the conformation of the integrin. They usually need to stick out and expose to their active site so they can interact with other ligands of the extracellular matrix. This is the active form. But in some cases, they will bend it. They hide their active site like this. This will be the inactive form. So such a bending or kind of straight out extended pattern is controlled by some intracellular signal. So, for example, this binding of those signaling will activate the integrin, so they will expose together to such an extended site. So under cryo-EM, okay, you can see this extended pattern or bending pattern. Okay, so the cryo-EM, this I would like to kind of discuss this. So in the cryo-EM, usually just put some solution. Okay, those are aqueous solution. So, but the integrated membrane protein, how can you image the membrane protein which contains the hydrophobic domain in the aqueous solution? That would be non-energetic favorable condition, right? So people kind of generate such a nanodisk structure, phospholipid nanodisk. They will accumulate some phospholipid, and also those phospholipid will kind of and use some alpha helix synthesized engineered protein to render the whole phospholipid in the form of a disk. We call the phospholipid nanodisk, and then incorporate the integrin. Then this integrin will be really happy to the aqueous solution, and then you can use under electron cryo-EM, you can image their structure here. So this is the band structure, but you can see it. But once you activate it, you will see those extension patterns. So by this extension and band, they can activate the new adhesion site and inactivate the old adhesion site and to trigger cell migration. Okay. So let's take a look at this video. The plan is activating group to interact with this ligand for blood formation.
Platelet adhesion and aggregation. In flowing blood, red cells predominate in the axial stream, while the biconvex disc-shaped platelets are marginated along the vessel wall, where they are well positioned to monitor the integrity of the endothelium. The normal endothelium provides a non-adhesive surface to circulating platelets. However, when vessel wall injury occurs, for example by cutting or separating a vessel, or as shown here by a puncture, and there is endothelial damage, the initial response of platelets is that of adhesion to collagen fibers in the exposed subendothelium. Collagen is one of the most thrombogenic components of the subendothelial matrix, responsible for the initiation of platelet adhesion. A number of adhesive receptors on the platelet surface membrane interact either directly or indirectly with collagen. Initial binding of platelets is considered to occur via the integrin alpha-2 beta-1 GP1A2A receptor, which allows for further binding of collagen via the GP6 receptor, initiating transmembrane and subsequently intracellular signaling. Adhesion of platelets to the exposed subendothelium is influenced by shear rates. At high shear, alpha-2 beta-1 and GP6 are not sufficient to initiate binding to collagen, and binding of the GP1B95 receptor to von Willebrand factor, abbreviated here as VWF, that is immobilized on collagen, becomes essential in platelet adhesion. Platelet adhesion at the site of vessel wall damage initiates activation events that result in aggregation. Adherent platelets undergo a dramatic shape change to an irregular sphere with multiple filipodia spreading on the subendothelium, increasing their area of surface contact. Adherent platelets also secrete or release the contents of their storage granules, the alpha and dense granules, by an exocytic process. This provides a high local concentration of effector molecules essential for platelet plug formation at the site of vascular injury. For example, the aggregating agent ADP is released from the dense granules. Platelet activation stimulates the formation of another aggregating agent, thromboxane A2, abbreviated here as TXA2, via the arachidonic acid cascade. Details are shown in figure 26.5. The most potent platelet aggregating agent, thrombin, shown here as factor 2A, is formed via the coagulation pathway from prothrombin, or factor 2, on the procoagulant surface of the activated platelet. Details are shown in figure 26.6. ADP, thromboxane A2, and thrombin bind to specific platelet membrane receptors and stimulate aggregation on and around the platelets adherent to the subendothelium via receptor-mediated signal transduction events. Aggregation is an active metabolic process. Binding of any of the agonists to their respective membrane receptors initiates signaling pathways that ultimately convert integrin alpha-2b beta-3 or GP2b3A from a low-affinity resting state to a high-affinity activated state for binding extracellular soluble ligands such as plasma fibrinogen and von Willebrand factor. Fibrinogen is shown here. The transmission of an intracellular signal leads to disruption of the complex between the cytoplasmic tails of alpha-2b beta-3, followed by a conformational change in its extracellular globular head domains from a bent to an extended state, promoting the binding to fibrinogen and von Willebrand factor. Divalent fibrinogen and multivalent von Willebrand factor function as bridges between alpha-2b beta-3 receptors on adjacent activated platelets, thus allowing platelet aggregation to proceed. In this way, the large and complex metabolic repertoire of platelets allows them to effectively perform their primary physiological role, that of supporting hemostasis upon tissue trauma, to form a platelet plug that arrests blood loss from a vascular injury.
Okay, so we just quickly summarize the key step. So once the blood vessel injury happens, the collagen will expose to the blood vessel, right? The lumens and those platelet in the blood, those will kind of expose to those collagen. So collagen will activate those platelet and trigger some signaling, downstream signaling and change the platelet activity. So it will activate some integrin. So the integrin will interact with the other platelet integrins through the fibrinogen. It will kind of bridging those collagen. And so integrin. So those are different platelet can form the kind of thrombus here to prevent the blood vessel leakage. Blood leakage. Okay. So this is triggered by the integrin. Okay. So let us move on to the Ig domain. So the Ig superfamily contains a lot of Ig domain. So basically, that domain is a common protein domain. Was first identified in antibody. So that is why we usually call it IgG, IgH, IgM for our antibody, right? So those Ig domain actually is a common domain and also found in this different camp. Okay, so depends on where they found or where they measure function. So neuron will be in neuronal camps, which mainly in the neuronal cell, or in the cell camp will be ICAM. Okay, it will evolve in the leukocyte into tissue, which we will introduce later. And the junction of intermolecular gems will exist in the titration. That is one of the camp we introduced in the titration section. Okay, so the important part is those induction is not calcium dependent. They don't need the calcium. Category need calcium. Ig domain don't need calcium. Okay, they interact without it's independent calcium independent pairs. Okay, and such an Ig interaction is important for the leukocyte internalization. Okay, so a lot of immune cells they keep traveling in our circulation in our blood flow, but once we need the kind of trigger some immune response or try to against the pathogen in the local tissue, we need to stop, ask those immune cells to stop, and then penetrate our blood vessel to the tissue to deal with such an immune response. Okay, so this reaction, this process we call leukocyte actualization. Okay, yeah. So let us take a look at this video.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. E-selectins on endothelial cells interact with ESGL1, a glycoprotein on leukocyte microvilli. Leukocytes pushed by the blood flow adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. At sensitive inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G protein-coupled membrane receptors. This binding causes the conformational change in the cytosol displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the 4 steps of a process called leukocyte externalization.
Okay, so this is the graph from textbook. So on the steady state, there will be no endothelial cell on the inactive state. So, and although the leukocyte contains several inactive integrins and several carbohydrates, they kind of don't have a strong an affinity with those endothelial cells. But once those endothelial cells be activated, it could be the immune response or could be the some inflammation around here. It will trigger the expression of the PAF, which is the plasma activated factor here. This will expose to the surface of the endothelial cell. And it will also transfer the vessel containing P-selectin onto the surface. So the P-selectin will bind to those carbohydrates on the leukocyte. So the central binding will activate the integrin. So integrin will become the activation. But the affinity between those PAF and the PAF receptor and the P-selectin and those carbohydrates is weak. So it will not totally stop, will not totally completely immobilize the leukocyte. It only gradually slow down the leukocyte. So that is why they will call this step. It's a rolling. Okay, so it's just gradually rolling and slow down. And when changing their angles, the activated integrin will have a chance to enter with the IK2. Okay, so or IK1. So it will form this really strong interaction. Then it will totally stop here. That will trigger such a trans-endothelial cell migration. Totally, this whole space process we call it ectropization, leukocyte ectropization. So it will go to the lower, deeper tissue to trigger some immune response there. Okay, so this is really triggered by the ICAM and the integrin interaction that I'm trying to introduce here. Okay, so and also we keep claiming that interaction between the cells and extracellular matrix is important, and also those junction or those membrane protein need connect with some attachment protein and they need to conjugate with some cytoscape. Okay, this will be the case to show why it's so important. So dystrophy is one adaptive protein connect with the membrane component. This membrane component complex will connect with the laminin or extracellular matrix, okay, of the muscle cell. And dystrophy is an adaptive protein will connect this complex with the active film. It can also stabilize the whole complex together. Once this dystrophin have a mutant or become either malfunction, this complex cannot enter with cytoskeleton anymore. Okay, and it will be easy, and it will also lose their interaction with the low extracellular matrix. And then the whole cell will kind of keep moving when we are, this is muscle cell, when we move, do the exercise, those cells will keep kind of have operations of the extracellular environment. Then the membrane will damage what happens, then totally this cell will kind of don't have intact membrane and finally will shrink it or die. Okay. So dystrophy mutant finally will cause one kind of disease called Duchenne muscular dystrophy. Okay. This just an example to show you how this adaptive protein, why this adaptive protein is so important for this for physiological condition. Okay. So just quickly summarize ESP, the transition between the non-motile epithelial cell and motile mesenchymal cells. And then motile cells integrate the form of adhesive structure. And such a two conformation of the integral low affinity inactive banded one, and the high affinity is extended one, straight out one, will be kind of related. The plasma activation integrate to form the clot. Okay. In the CAM, we just talked about that there is several CAM can be found in the different tissue, And those IgCAM and integrated interaction is also important for the leukocyte vectorization. Any question? Okay, so let's jump to the final section. So after introducing so many different animal stuff, now we shift to the plant cell. Okay, so the plant cell, this is the one plant cell, this is the process membrane, the membrane plant cell. They also have some extra matrix protein, which is cell wall. Okay, so they will be the primary cell wall and the secondary cell wall, and some middle lamina. Okay, basically they do the same thing. They just kind of form another binding site, and they can form the diffusion barrier, and they can also help glue two plant cells together. Okay, so for there are several components, for example, the cellular microfibrillary, which is a long chain of those are glucose, okay, and the pectin, okay, so which is kind of combined with the water and kind of can group different plant cells together, and the hemicellulose, which is a branch polymer of 5 or 6 carbon monosaccharide. So the cell wall have a lot of sugar, okay. So after the cell died, people will collect the cell wall because cell wall is super strong, so they will retain even the plant cell die. So and they can dissolve those sugar to generate more ethanol. They will be the kind of biofuel. Okay. The people use those to generate biofuel. Okay. So and also animal cells have AMP enzyme can digest BCN to generate space for them to do some activity, but how about the plant cell? Okay, so they need to loosen the cell wall to generate more space. So now just give this as an example, phototropism. Okay, this is an event that the plant will grow toward the direction of the sun. Okay, so how plant can achieve this? So there is a really important plant hormone called auxin. So auxin will accumulate to this is the plant. Auxin will accumulate to the other side of the sun, and those auxin can activate once proton pump, which can be activated by the auxin. So once you have accumulated more auxin, it can keep pumping proton to the interstellar space and make this environment more acidic. Then those cell will are losing. Okay, the plant cell cannot migrate, but they can accumulate more water here and then extend their vacuum. That is the way they kind of extend their cell size. So by doing this, by increasing the cell volume from this side, okay, so they will have growth toward this, will bend it, the whole plant and face to the direction of the sun. Okay. And also, the one cell junction only happens to the plant cell is the plasmodesmata. This is really amazing structure. So this is one plant cell, the other plant cell, they have same kind of connect with each other. They ER and the plasma reticulum connect with each other. Okay, and the lumen of the ER will connect with each other, and also cytosol also connect with each other. Okay, so we call this a cytosol part of annulus because the section we'll call the space here we call the annulus. So basically, for the plant cell, they don't have identity because they share the same cytosol environment and they also share the same lumen of the ER. So they basically, this A cell and the neighboring cell, they don't have a strong identity. And for the animal cell, we have recently we have a kind of one spatial structure called the tunneling nanotube, TNT nanobomb. It's a tunneling nanotube. Okay, it first described in the 2004. Okay, which is a membrane-based structure with a diameter of 50 to 200 nanometers. So it's basically it'll connect. It's a tunneling nanotube can connect between the two membrane cells. Okay, it basically can deliver anything. It can deliver organelle, vesicle, protein, charge, and it can even provide some. It can also deliver virus. If one cell infected by HIV virus, they will can kind of infect other cells through this tunneling nanotube. The virus can travel between the cells through this nanotube. Okay, and if you see, you can see here, those are tunneling nanotube. They are not right on the bottom. They actually is a skywalk. Okay, so you can imagine they kind of have a strong force between the two cells. They are not right on the bottom. Okay, and they can deliver a lot of things between this cell to the other cell. Okay, research only labels stuff here, so you will deliver regionally from here to the other cell. So people already know the P53 and the EGF signaling are involved in such a TNT formation. And then people curious about who will be the donor, who will be the recipient. Okay, and then somehow people found the unhealthy cell will be the donor. Unhealthy cell will generate will be actively generate the TNT now to connect to the healthy cell and then deliver stuff to those healthy cell. Okay, and the people who kind of found this, they have a hypothesis. They say because to share, if they know they are going to die, they share those material can kind of increase survival rate of the whole cell population. I think it's a bullshit. Okay, because when you publish something, you publish an article, I know you need to be positive. That is why your hypothesis is positive, but it doesn't make any sense. To me, it's like, I'm going to die. How can you survive living? So just share some knowledge to you. Yeah, that would be more make sense. But you cannot write this political incorrect stuff in your manuscript, right? Okay, yeah. Okay, so this is an image from my lab. So there will be two cells. Those are yellow signal mitochondria, and it's a tunneling nanotube. They can, even mitochondria, such a huge organelle, can move across a different cell through the TNT. Okay, so this will be the other side. Okay, so the final section we just introduce about the cellular key component in the full cell wall, cellular microfibrils, pectin and hemicellulose. And we also talk about the oxygen, how it can activate the proton pump to degrade the EC and the cell wall to facilitate the cell elimination. And we also introduce the plasmode desmata and also the tunneling nanotube to transfer material between the animal cells. Okay, any question? Although, if no, all of my tests will come from what I teach in the class. Okay, if you find anything I haven't teach in the class, I will give you score. Yeah. Okay, so basically I probably will have, you will have 6 or 7 or 8, I haven't decided, question, you need to answer that. Okay, yeah.
So is it like open questions or the direct?
Yes. It's a open question. Any questions about? No? Good luck for your test.